Good morning, grade 9 students. How are you all? So, are you taking care of this COVID 19? Yes, please stay home in eradicating this virus. Well, so uh, we have started some digital classes so order to not to miss the academic side. And of course, we have already started the class 9 and you had some practical sessions from the lesson fundamental units of life. So, let's recap it. The first one, what we observed is the parts of a microscope. So in these parts of the microscope, we have seen the eyepiece through which we see the smear that is placed on the slide, and this eyepiece consists of two types of lenses. One is the 10x and the other one is the 15x, and connecting to the eyepiece is the body tube. This body tube is movable and it moves up and down with the help of this cos adjustment knob. The other end of the body tube, we have a revolving nose piece. So this rotates, as you know. And objective lenses, there are three types of objective lenses: 10x, 45x, and immersion 100x. Right. So here is the cos adjustment knob, which helps in proper visualization of the smear. And this is called as the stage on which we place the microscopic slide, and the slide is held in position by using these pins. All right, then the condenser. This is the condenser which is placed over here. So this helps in regulating the amount of light passing through it, just like the iris of our eyes. And below that we have a mirror. So it is a digital one, so it emits the LED light. In ordinary microscopes like what we have in our laboratory, they have two types of mirrors. One is the plane and the other one is the concave. So you know under which light, which type of mirror we have to use. So in our previous session, we discussed about the parts of a microscope. From this session, let us learn how to make a microscopic sections or the smears using our vegetables. So let us see what are the materials required to make the sections. So first thing, what we need is a specimen vegetable, which could be any type, and we need a clean slides, then a scalpel or a dissection knife or a blade to make the fine sections of the vegetables, and a dye, a crystal violet or saffron in order to visualize the cells, and the cover slips to make or to mount the smear, a dropper to add the dye onto the sections, and the dissection needle. This helps us to place the cover slip over the slide. The filter paper, this helps us in removing the excess stain from the sections, and of course a microscope to visualize the cells, and a glycerin. This allows the smear from drying. Alright, now let us see the procedure in making the fine sections of the vegetables. First thing is we have to make a fine section cutting using the scalpel or a blade. Then on a clean slide, we need to add a drop of water and transfer this fine cutting on to the slide. Then a few drops of dye, which you have already discussed, either crystal violet or saffron is added to the cutting, and it is left undisturbed for a few minutes so that the dye enters the cells. Then the excess stain, after some time the excess stain is removed by using a filter paper. Then a few drops of glycerin is added to the section cutting, and using a dissection needle, a cover slip is also placed over the cutting, leaving no air spaces. So we have to ensure that no air bubble is formed between the cutting and the cover slip. Then the slide is ready for the observation. Now this can be placed under microscope. Then first, after placing the slide on the stage of the microscope, first thing is we have to adjust the mirror so that the light is passed on to the condenser. Then fixing the eye lens to 10x, we have to use the cos adjustment and see the place where we could see the cells, that is the visualization of cells in the section. Then on finding the cells, once we find the cells using this 10x, then the resolution of the objective lens may be increased to 45. If you have an immersion oil, further it can be increased to 100x. Next, after observing the cells, we need to draw the observations what we have observed under microscope in the record book. And the last thing, you should not forget to clean up the worked place. Whatever the cuttings of the vegetables or the fillings of the dye should be cleaned, and the lab has to be maintained neat. So here are the few vegetables we observed under the microscope. So this in our school lab. The first one is the beetroot. Second is the potato cells. The third one is the blood cells. Can you guess what it is? Yes, these are the matchstick cells and the cells from the lady finger and the onion peel cells. And students, can you tell me why we did not add dye while observing the blood cells? The structures what have we seen before were the cells, and from this you could understand that all the living organisms are made up of cells. So therefore these cells are considered as the fundamental units of life. So in every organism we find cells. So students, when every organism is made up of cells, I have a question for you. So till the next session begins, try to solve this question. When all the living organisms are made up of cells, why all living organisms do not have same shape and size? So till we catch up in the next session, try to solve this question. Have a good day.